Hi everyone, welcome to ABio 365 and Biological Chemistry 1 or Biochemistry 1 for the fall 2020, and its class number is 1322, and the lectures are going to be online, unfortunately due to the COVID-19, but the recorded lectures are going to be available on the Blackboard. So my name is Chang-Hwan Lee, I'm an assistant professor of the Biological Sciences. And you can find my contact information right here, especially the email address, which you'll use when you have any questions. And when you do, please use Bio 365 in the subject line. And we'll also have office hours on the Wednesdays and Thursdays from 1 to 2 p.m. using the Zoom meeting. And the access information is right here, and we're going to use the same information here every time we have office hour. So the textbook we're going to use is Fundamentals of Biochemistry 5th edition by Voigt and Pratt. And the readings are going to be listed on the lecture schedule on the syllabus. So please refer to that and read those sections accordingly. And these readings are going to be considered as an assignment, although there are not going to be any points for these assignments. Lecture presentation, including videos, slides, and/or notes, will be available on the Blackboard. Here's the address. And all these are copyrighted, so please make sure not to modify any of those materials or distribute them. So we're not going to have any physical attendance because all the lectures are basically pre-recorded and uploaded on the Blackboard. And all those lecture videos will be available per the lecture schedule on the syllabus. And you're the one who's responsible for any announcements made during the recorded lectures or on the Blackboard. And that includes the exam date changes and some of the critical changes too. So please check those Blackboard or watch the recorded lectures every time and regularly too. And the students must receive the written permission from me, basically, of the instructor, if you want to use any recording devices or apps during watching the lectures. So you'll take 3 exams in total, and each exams will only cover the materials presented after the previous exam and before the current exam. And all 3 exams will be considered equally for the final grading. Most importantly, the exams will be taken on Blackboard. So unlike in-person classes that you just walk in a classroom for taking the exams, now you need to actually go to the Blackboard, and there's going to be a module available on that particular time that was scheduled for the exam that is open, so you can access to that. And you're going to have much more information once the date's closer comes closer to the exam date. And during the exam, you'll not be able to or allowed to use textbooks or the lecture presentations and or online materials. So we continue on the testing policies. Most of the questions are going to be in a multiple choice format, although it can include some multiple logic steps or the additional essay type questions too. And some questions might involve calculations, but you're not going to need a calculator there. And this is important information, so I'm going to just read it aloud. The only individuals allowed to take a makeup exam are those who missed their normally scheduled exams with the legitimate reasons established by the university. And you can find the form right in this link. And that means that you need to actually receive a memo from the Office of the Vice Provost for Undergraduate Education to make an excuse for that. And in case you made an excuse, then the missed exam will be taken at the second hour of the final exam period. So we'll set up on time for that too. But this makeup exam will not contain the same questions. Now move on to the grading. Each exam has a total of 100 points. and the final letter grade will be derived from the average of all the exams. But any missed exams will be graded as a 0 point, and missing all 3 exams will make an F. And this is the grade scale and the conversion, so you can actually take a look. So what is biochemistry? Of course, it's a biological chemistry, which is a combination of biology and chemistry. So we try to understand biology in the terms of chemistry. So a bit more elaborate definition of the biochemistry is the study of a life on a molecular level, seeking to describe the structure, organization, and function of living organisms in molecular terms. So the biochemists are concerned with everything, including the organic stuff and inorganic stuff too, which can affect those living organisms. So to better understand the life on the molecular level, you must understand these 3 things. First is the chemical structure of the biological molecules, so you can better understand how these molecules interact with each other. And the second one is the biological function of the molecules. Of course, you want to know what these molecules do. And lastly, the bioenergetics, so you can better understand the energy flow within the cells, within the tissues, or the organism. And the biochemistry is also a research discipline applicable to the real world, not just study on the desk, because all those findings from the biochemistry can be applicable to the agriculture, so make the better crops, more efficient cropping too. And also the industry for the mass production of the biomaterials and so on. And also the medicines for the better drugs targeting the more effective reasons of the particular disease or the virus. So biochemistry, let's talk about it in the dramatic narrative. So the characters and roles are going to be the actual biomolecules and their structures and functions. And the stage is going to be basically water, because the cells constitutes more like mostly water, over 70%, around the 70%. And that could be the medium of all the molecules reactions. And the major story lines is going to be the flow of the matter, information, and the energy, which all going to be governed by the law of thermodynamics, because all the molecules are working in the certain environment. So there are 3 areas of studies for the biochemical field. And these are the common themes in biochemistry. First one is informational, so which studies about the language for the communication inside and between cells, as well as the between cells and the environment. And conformational study will consider the structure and the 3D arrangements of the biomolecules. And lastly, the metabolism is going to be studying about the energy production and utilization. And basically, the biochemistry is going to be the study of the structure of the biomolecules and make a connection with their functions using the term of the bioenergetics. And then you're going to get the will be able to describe the chemistry, what is really happening within the living cells. So this is the tentative schedule syllabus for the Bio 365 in the fall. As you can see, you can take a look more closely, but there are going to be 3 exams, basically consists of 2 midterm exams and the final exam. And in between them, we're going to study about the thermodynamics, which I told you that which governs the biomolecules, reactions, and functions. And then water, which is critical for the any kinds of biomolecular reactions, and then basically their constituents: nucleic acids, proteins, carbohydrates, lipids, and also lipids makes the biological membranes. and then enzymes, the proteins, and also metabolism, how the energy flows within the cell. So this course, of course, cares about biochemistry. And again, we want to understand the biochemistry is really a field of science that investigates the chemical and molecular reactions that sustain life. So we are going to focus a lot on this chemical and molecular reactions during the course. And the goals for this course is that first, you're going to study the chemistry and biochemical interrelationships of the nucleic acids, proteins, carbohydrates, and lipids, as well as enzyme catalysis and metabolism. And we're going to, touch basically all the bases of all these different molecules and the reactions. And then after that, you must be able to describe the chemical structures, properties, biological functions of molecules, of these different molecules that make up the living matters. And lastly, you should be able to comprehend the current biochemical literatures. So what are the biomolecules, or the even cells and organisms? Let's start with the atoms. So there are, of course, atoms in the universe everywhere, and that actually makes up the molecules, and all the molecules gather up to make the macromolecules or micromolecules, and these are actually the biomolecules. And then when those little proteins or the macromolecules get together, then it forms the organelles, which are basically subparts within the cell. And of course, little subparts get together to form the cell. Multiple cells gather up to become a tissue. And these tissues with the different types of cells get together to form the sort of independent large organ, which has a specific function within the organism. And these different organs makes the whole body, which is a single organism, independent system that can live using the flow of energy. And of course, at the end, getting all these organisms get together to make the society or the population. So biochemistry cares all about these, but particularly we're going to focus on these little small molecules, including atoms, molecules, and macromolecules, to see that how they interact with each other to actually make all the functions up to the population. So let's quickly talk about the structure of the cell, which is the critical part in the biology. So regardless you study the biochemistry, molecular biology, or cell biology, you need to understand precisely what you're looking at within the cell and how this cell is formed. So of course, a cell has typically single nucleus, or it can be multiple nuclei at times. Anyway, this nucleus has the nuclear membrane and nuclear envelope, which actually separates this nucleus from the cytoplasm. And nucleus also contains the nucleolus, which contains a lot of important proteins and other RNAs critical for the transcription within the nucleus. And outside of the nucleus, and of course, the critical thing is the DNA itself, the chromatin, or the multiple strings of the nucleic acid. And then outside of the nucleus, there is an endoplasmic reticulum, ER, and they have two different classes, which is rough ER and smooth ER. Rough ER is close to the nuclear envelope, which is responsible for production of proteins, and the smooth ER is for making lipids like steroids. And there are also free ribosomes that are floating around in the cytoplasm for the translation. And there's also a critical part within the cell called the mitochondria or mitochondrion here, which is responsible for the respiration of the cell. So it produces the energy by using the oxygens. And also there are lysosomes, which is responsible for degrading some proteins and some other parts within the cell, and vacuoles basically a storage of the cell, and the Golgi apparatus responsible for distributing the cells in proteins in the proper places, and centrioles for the cell division. So when we look at the biochemical compositions of the cells, it's, of course, all of them are just chemicals. So that's why we need to, better understand what the chemicals, those biomolecules are, and how they interact. And of course, the biochemistry itself is the study of this chemical stuff, and it's chemistry in living cells. When we look at the composition of the cells, of course, about the 70% is just water, and 5 to 10% will be small organic molecules like vitamins, fatty acids, sugars, inorganic ions, etc. And the rest, 20 to 25%, be the macromolecules. And all these molecules are needed for maintenance and perpetuation of cells and to coordinate cellular activities. And there are about 100,000 different types of molecules in just one animal cell. It's not the total number of the macromolecules or the micromolecules. It's actually just the types. So there are lots and lots of chemicals within the cell. And the small molecules are generally imported into the cell through the membrane. So it's more like by the diffusion you can take up, or you can actually actively just engulf those small molecules. And these small molecules are going to be used for synthesizing the macromolecules within the cell, because macromolecules are really the functional units, but it's hard to intake by the cells because of the size and also the stability and the availability in the environment or outside of the cells. So the macromolecules are the most interesting and characteristic molecules of living systems because they can be easily formed and also degraded, and also they have all their specific functions, which gives a lot of a variety of the in terms of the functions of all those cellular components. And macromolecules are large polymeric multi-subunit structures made up of smaller subunits called monomers. So monomers works as the little Lego pieces, and then they can build up to the much bigger, higher with the higher structured multi-subunit polymers. And the examples of macromolecules include DNA and RNA, which are formed by the little pieces of nucleic acids, and these little proteins, and also including enzymes and antibodies formed by the amino acids, and also carbohydrates from the sugars, and lipids from the fatty acids. So the cells virtually contain just 4 major families of the small organic molecules to synthesize such macromolecules. First one is nucleotides, which are subunits of DNA and RNA, and that is for storing and transferring the information within the cell. And second one is amino acids, which are the subunits of the proteins, and these proteins are the workhorse of the cell. And simple sugars are the subunits of carbohydrates, and these are going to be the food or the energy source of the cells. And then lastly, the fatty acids are the components of the cell membranes, and also subunit of lipids, and the lipids are the storage of the cell basically for the excess energy. When you look at the size of these macromolecules, the nucleic acids forms the DNA, and it can be up to 10 to the 10th power of the subunits, so it can be super big. And also carbohydrates, lipids, and the proteins can consists of up to 10 to the 4th of subunits, their own subunits, so they can be quite big too. And the shapes can be globular, helical, hairpin, and or fibrous forms, and also can many other specific forms there. So to better understand the biochemistry, you need to understand the 3D structure and the architecture of those macromolecules. because once you see the structures, you can see that where these two molecules or multiple molecules interact, so-called interfaces, and you can better understand how they work. So it can give you an idea for the function, and also the function itself can give a better clue for the protein-protein interaction in 3D. Now let's look at the chemical composition within the cell and within an organism. So the majority of macromolecules contain only 6 atoms here, which are the carbon, nitrogen, oxygen, hydrogen, phosphorus, and sulfur. And you can see that the percentage is pretty big here, especially the carbon. And to stabilize the macromolecular structure, the chemical bonds and the other interactions are needed, because these the atoms basically cannot just stay together in a small regions and just stay like that for a long period of time for the actual function of the macromolecule. So they utilize the covalent or non-covalent bonding. And non-covalent interactions are quite important for the interaction between the macromolecules. And these interactions include hydrogen bonding, ionic interactions, hydrophobic interactions, and so on, and which we're going to discuss a lot about at the beginning of this course. So what does biochemistry study? It studies chemistry of biologically important molecules, including biomacromolecules. Okay, then what does chemistry study? It studies composition of matter, its changes, and its chemical and physical properties. So biomacromolecules also have physical and chemical properties, and its physical properties can be revealed without changes in chemical compositions, because you can just put down the macromolecules and examine it. But the chemical properties has to be revealed only through the changes in chemical composition. And there's a saying, structure is function, or structure determines function, which means that this structure actually determines the chemical properties of the macromolecules, which result in its particular functions. This chemical properties of element can be determined by the atomic structure, and the chemical properties of the substances can be determined by the molecular structures, of course. And the chemical property of the macromolecules will be determined by the 3D structure of those macromolecules, such as proteins and the DNAs. So covalent bonds forms the structure by put together the atoms in macromolecules or even little chemicals. And I know that you've learned the covalent bonds as well as some other type of bonds in organic chemistry or other chemistry classes, but we're going to just cover some basics here again and revisit them. So first, the covalent bond is the most stable bond type. And Gilbert Lewis in 1916 has proposed that the atoms transfer or share electrons in a way to attain filled valence shell structure, known as the octet role, because it takes the 8 electrons to achieve that. And that is the guiding principle of how the atoms are bonded. So here are the major atoms or the elements in the macromolecules. And these black dots are the electrons on the outermost shell in the orbital. And each type of atoms form a characteristic number of covalent bonds with their atoms, because it depends on how many atoms that are available on that outer shell. So for example, carbon can make 4 bonds using these electrons, as opposed to nitrogen or phosphorus can form the 3 bonds here, and so on. And the covalent bonds have the shortest bond length and the strongest bond, because it requires a lot of energy to break. So the thermal energy, for example, at 25 degrees C in cell is the 1 kilocal per mole or kcal per mole. But the carbon-carbon bond requires a 348 kcal per mole to break. So it's a lot of energy. And the most of the biomacromolecules are carbon compounds because of the propensity of carbon that forms the stable covalent bonds with other atoms, and it has the most valency as 4, so it can form the 4 bonds. And there's a propensity of carbon to form the stable covalent bonds to itself too, which is called catenation. And this carbon, because it has 4 atoms available, it will form the tetrahedral structure, which will give the another property, chemical property, such as directionality or the handedness. And there are some important functional groups in biology, so I want you to remember and memorize all these basic functional groups so we can talk about it over and over again without revisiting these actual structures. So there are alkane, alkene, alkyne structures defined by the carbon bonds types. And the alcohol includes the hydroxyl group. And the ether is basically have an O linkage. And the amine has the amino group or the nitrogen with the hydrogen group. And the thiol has the sulfol. And the aldehyde have the CHO group right here. And the ketone has the CO in the middle. And the carboxylic acid has the carboxylate group. And the carboxylic ester has the ester bonds right here. And the amide, of course, has the amino group. And the phosphoric ester and the phosphoric acid anhydride had all the phosphorus in their groups. So in contrast to the covalent bond, which is the strongest bond, there's a weak interactions happening in biology too, and it's really important, especially in biochemistry, due to their sheer number. So there are a lot of weak interactions happens within the cell, and that is also responsible for inner and intramolecular attraction and interaction. For example, it holds the DNA strands together, and it also helps the protein folded into a tight globules, and it's a specific structure too. And these bonds can break and reform pretty freely at room temperature. So it's a transient bond as opposed to the covalent bond, which is really tight and cannot change too easily, especially at room temperature. So now look at the bond energies. The covalent bond ranges from 200 to about 800 kilojoule per mole, which shows a really high energy, as opposed to weak interactions reside right at the bottom of this diagram. And among the covalent bonds, especially this range with the CC bond or CH bonds, is the most biologically relevant covalent bonds, because these bonds are really strong enough to be stable, but also not too strong as to be impossible to break. And also these weak interactions are biologically relevant, which is continuously broken and reformed, and that is important for the function of the biomacromolecules. Now let's move on to the first topic, thermodynamics, part 1. Thermodynamics is the study of energy and its effect on matter, with the Greek words thermo meaning heat and dynamics meaning power. And definition of energy here is the capacity to do work or the potential to transfer heat. And the definition of work here is something has done, and it can be described in many different ways, including mechanical work, W equals F times D, force times distance, or pressure volume work, which is PV, that it applies to the gas reaction, and also other types of work, including electrical work, BQ, and so on. And both energy and work are measured in joule, J, or cal per calorie. And 1 cal is 4.184 joule, but the nutritionist actually use 1 cal with the capital C, meaning that that's 1,000 times of the cal, so 1 kcal, kilocal, which is the same as the over 4,000 joule. So why do we study thermodynamics, especially in biochemistry? Because thermodynamics can tell us whether a given process will occur spontaneously or it will require energy input, and if that's the case, then how much? And then how much is the same as expenditure of the cell for making a certain molecule too. So you can better understand how the macromolecules interact with each other in the cells, and also some signaling pathways or some metabolic pathways, how the molecules change with the expense of their energies. However, thermodynamics can tell you basically nothing about the rate or the speed of the process, because this rate or the speed of the reaction depends on its mechanism, as well as it's subject to more like kinetics rather than thermodynamics. So let's talk about the definitions relevant to thermodynamics before we delve into the thermodynamics details. First, a system is a part of the universe under study that you're interested in. The examples are the organisms, cells, reaction tubes, etc. And the surroundings basically is everything else other than the system, so rest of the universe. So when you look at this green region, let's consider that as the universe, the whole thing. Then this tube region or the space is going to be your system, and you're looking at something inside of that and their energy changes, and something the system outside of this system will be the surrounding. And there are largely three different types of systems. First one's isolated system, where there's no changes of matter or energy at all with the surroundings. And closed system is the one that exchanges only energy, but not the matters. And the open system is basically change everything, including matter and energy. And all the organisms, the cells, and our living systems have this open system. So again, thermodynamics is the study of energy and its flow and its effect. And it has a few laws. And the first law is that the energy is conserved and cannot be created or destroyed, meaning that they don't make it anymore. There's not going to be any brand new energy generated. But it can be converted from a form to another, which means that the total amount of energy within the system and the surround, which means the whole universe, is constant. There's an exception, E equals mc squared by Einstein, meaning that the energy can be generated from the mass itself. But we're going to ignore this discussion because it happens only in the sort of extreme cases, like a black hole. A more useful example is going to be ATP to muscle contraction. So ATP is a molecule that stores the energy in a chemical form. And then if the energy is converted to the mechanical form where muscle moves. But it's all about the energy form conversion, not about the creation. So there's not going to be any free lunch or anything generated. So energy is a state function, which means it depends only on the current state of the system, not on the way in which the system got to that state. So it doesn't care about the past or future. It only considers the current state. Let's think about a skier that climbs up to the top of the hill, or another skier that could be taking the lift to the top of the hill, or deposited by the helicopter. As long as they're on the same position from the ground, they're going to have the same energy or the energy potential. And energy change can be written as delta U equals delta U final minus delta U initial, where U means the energy or internal energy, and delta means the difference. And of course, that's the final energy minus initial energy. And this U is the grand total of all forms of energy, including kinetic energy of the molecules when they're moving, as well as energy of attraction and repulsion of the subatomic particles, atoms, ions, molecules, and so on. And this delta U, which was U final minus U initial, can be also written as Q minus W, where Q is the heat given to the system by the surrounding. So it's basically the energy that the system gets. And the W is the work done by the system on surrounding. So this is the energy that the system loses. And here's the sign conversions that can be useful. When the Q is larger than 0, that means the system absorbs the energy from the surrounding. So it's an endothermic process. And when Q is negative, that means system gives up the heat to the surrounding. It's an exothermic process. And when W is larger than 0, that means the system does work on the surrounding. And the W is negative, then that means the surrounding works on the system. So remember, work is something has been done, and it can be described in many different ways, including the product of force and distance. But the work that is most relevant to the biochemistry is the pressure volume work, or PV. And the changes of these vectors can be written as delta P, delta V, or delta PV with a parenthesis. And biological reactions typically takes place at constant pressure as 1 atmosphere, so it can be written as P delta V instead of delta PV. And the only other form of the work that we're gonna talk about later classes is gonna be the electrical work, which is VQ. So now let's define another critical state function for thermodynamics, enthalpy, or H. It's a total heat content of a system, and H equals U plus PV, where U is the internal energy and PV is the work. And the changes of enthalpy can be written as delta U plus delta PV. In cells with a constant pressure, which is the most cases, delta PV is almost the same as P delta V. So we replace this delta PV with P delta V here. And then the formula is going to be like this. And then we put the expression for delta U2, which was delta U equals Q minus W. So instead of delta U, we put the Q minus W here to get delta H equals Q minus W plus P delta V. And the QP here is the basically Q, the heat, at a constant pressure. Again, bring up the previous formula here, delta H equals QP minus W plus P delta V. But because the most relevant form of the work is PV, so we can actually replace this W with the P delta V. So replacing by this and canceling this P delta V here that was already exist, then it's gonna end up having delta H equals QP. And that means the changes of enthalpy is almost the same as the amount of the heat absorbed in a process with the constant pressure. And when the delta H is larger than 0, that means heat is absorbed and the reaction is endothermic. And that is basically enthalpically opposed, because usually the enthalpy, the energy, is emitted out to the environment. If the delta H is negative, that means the heat is given off, so it's an exothermic, so it's enthalpically driven process. In biochemical reactions, usually delta V is also 0 because the size of the cells or the molecules doesn't change that much. So you can actually apply 0 for here, then it's going to leave the delta H equals delta U. So internal energy changes is going to be enthalpy changes too. So can delta H or enthalpy change tell whether a reaction is spontaneous or it requires energy input? Again, the spontaneous process is the one that proceeds towards the state of lower energy from the high energy. Let's look at the example here. In the ice bag, there's a reaction happens where urea is dissolved, and that is an endothermic process, which means the delta H is positive. Thus, this reaction absorbs the energy or the heat from the surrounding, which is enthalpically opposed. But why does it occur spontaneously? Because it's not about the enthalpy or enthalpy changes that determines the spontaneity, but it's about the entropy process, which is another important state function of the thermodynamics. And we're going to talk about the entropy and enthalpy and the relationship for the spontaneity of the process in more depth next time.